Hello and welcome to the fantastic world of microbiology. I am Dr. Praveena, Professor of Microbiology and currently in charge at Sree Balaji Medical College and Hospital. Today we'll be seeing the secrets of microbiology. It will be an introduction session. So, according to the new competency-based medical education, this is a new curriculum, and today we'll be seeing competency MI 1.1. And before we begin, I want to tell you that it's a very interesting subject with lots of stories. There's actually a story behind every other microorganism, one that you know and you're familiar with. And the currently world famous microorganism is COVID disease causing one. That's a virus, coronavirus. This is for you to be familiar with it. Like this, we have plenty of bacteria, viruses, which we are going to see today. The specific learning objectives for this class will be the branches of microbiology, methods of study, and historical aspects and the road ahead. When we talk of the branches of microbiology, we have bacteriology, virology, mycology, immunology, and many other expanding branches. So what we are studying today in this session is medical microbiology. See, when you talk of microbiology, there are microbes, which are microorganisms present all over in the world. It's ubiquitous, meaning it's present everywhere. These teeny weeny microorganisms. But we will be interested only with those microbes which cause diseases in humans, which constitutes medical microbiology. So when you read, you can take class notes, or you can refer to your textbooks of standard textbooks of microbiology, and then we have practical sessions where you have hands-on training experiences. So it will be a very interesting course to do, provided you apply what you read. And then we'll be seeing about the historical aspects of microbiology, from the science that it was then to how it has evolved and developed into the technological advancements we have now. And the road ahead there are lots to be learned. It might be a bumpy road when you may have hitches, and we are always there to help. And you may have a smooth sailing with your friends and with your textbooks and with your passion for studies. Now, when we have the story time in microbiology, it actually has all those interesting anecdotes throughout the textbooks of microbiology, how things came into play. People in earlier days, they did not know about the existence of microorganisms. They thought that diseases were caused because of some sins, their parents' sins, or curse from God, or many other like. They did not know that microorganisms caused many diseases. That was the source of their disease. They did not know. So you have to rewind to a period in history where these information that you are having now with TV, media, and the like were not at all available, and people were so ignorant to the fact that microbes were causing disease. So, this session is a period classic where you really need to go back in history and understand it from that perspective. Now, medical microbiology is actually the science of microorganisms, that is, living organisms that are only visible under the microscope. So, this is for ease of study. It has been classified into, like I said earlier, bacteriology, which is the study of bacteria, like if you're familiar with those typhoid causing bacteria, salmonella, and virology. Now, the world famous or rather notorious viruses that we have, the coronavirus causing COVID, the HIV virus causing AIDS, this and many other viruses like those that cause chickenpox. These study of these viruses is what constitutes virology. And then mycology is the study of fungi, those that cause diseases in the skin, like for example a tinea infection in the face, disfigurement, discolorment, which cause and many diseases which infect the skin, hair, nails, and people. Every time any patient may be have experienced such fungal diseases. And immunology is the study of immunological reactions. Immune diseases are there, and even diseases have many immunological reactions, and the methods of identifying, like so there are certain antigens and our body produces antibodies. The study of that is immunology. So, medical microbiology, we will see the causative agents of all these organisms which cause diseases. Those diseases are called as infectious diseases because they cause infections. So, when you come to microbiology, this is a diagnostic science, meaning we find out what is the cause of this disease. It's almost detective work that you do. Investigate and find out the cause of any infectious disease. So, what you need to concentrate and how you need to study microbiology is you have to know the proper microbiological name for any organism, both the genus name, species name, and then its further subtypes. Without any spelling errors, you need to know the names. And then what disease it causes. And also what is the route of entry, how it enters into the human body, and what are the methods how it causes diseases. That is called as pathogenesis. How it enters into the body, what are the changes it brings, that are the clinical symptoms it produces. So how the doctor will interpret those symptoms. And then you have a history. So you really need to know that your mind does not see what your eyes your eyes do not see what your mind does not know. So you really need to be armed with this information, how it causes. And then the lab diagnosis, where you really need to know how it looks like under the microscope, whether it is possible to grow these organisms, that is called as culture. Whether certain organisms, microorganisms, you can grow in culture, certain that is in the lab, an artificial media on a petri dish, you can grow it. Certain organisms you cannot grow, and how you will be able to interpret all these diseases. And there are immunological diagnostic methods and many advanced techniques in diagnosis, like the molecular level, like PCR and others, for example. So all these diagnostic methods is what you really need to concentrate and study, the lab diagnosis. And then you also need to know how certain diseases can be prevented. Only if you know the pathogenesis, you can know where to halt it. So, and briefly about treatment as well, and vaccines if they are available, which is the mainstay. You know, by now, how many millions of people have been saved by vaccines. The one raging disease, the trending disease currently in vogue now, for example. So, this is what microbiology is all about. Now, if you look at this slide, you can see a person is examining clothing material. So there was a person who was studying the texture of this material. The person who does this is called as a draper, and this is in olden times. And he was interested in it, and he wanted to look at it better, and he was using a technology which he discovered. He was grinding lenses actually, and you can see he used to look at the quality of the cloth. That draper, he looked at the quality of the cloth, and then out of interest he began using those lenses to look at anything and everything under the sun. For example, grasses or water and all this. Please remember, this is set in the early much in the 16th century. So this person's name was Anthony von Leeuwenhoek. He was a draper in Holland. So what did he see in those other water and other samples that he took? He was surprised to see tiny bacteria, teeny new organisms. But of course, at that time, he did not know that they were living organisms. Then, out of interest, he drew them on a piece of paper and wrote about it and communicated to the Royal Society of London. So all these organisms which he had seen are now retrospectively we know them to be many bacteria, and they were the causative agents of many diseases. Please remember, it was at the time when no microscopes were there. So he is almost the first person to make microscopes with a single lens, and that one led to another, and so today we have such advanced microscopes thanks to Leeuwenhoek. The next great scientist is Louis Pasteur. You'd have heard of Pasteur's Institute in Paris, and in our own Tamil Nadu, in Kunur, there is a Pasteur Institute named in his honor. So what did this person do? How come he is so famous, Louis Pasteur? If you look at these two equipments, autoclave and hot air oven, they are very important sterilizing equipments. Sterilizing means to kill any living microorganism. So, for example, if you go to the doctor for an injection earlier days, or if you are going to an operating theatre which has knives and scalpels, all these instruments should be cleared of any bacteria or any living microorganism. So, to make it sterile, he devised the use of autoclave, which operates under pressure, and hot air oven, which operates under dry heat. So what did these two equipments do? They killed those bacteria and made the equipments like knife and scalpel sterile. So anybody operating, any surgeon who uses these to operate will be certain that it is a really clean, sterile knife which will not harm the patient or cause any infectious disease. So now Louis Pasteur, this person was a Frenchman who lived between 1822 to 95, and he was a chemistry professor, and his interests were fermentation. And he used this knowledge to for the good of fermentation. In the food industry also he celebrated, and as you know, he has discovered or invented all these equipments like the steam sterilizer, hot air oven. And then he went on to study about diseases like anthrax, the Bacillus anthracis, which causes anthrax, which is a disease in animals, chicken cholera. And you know what hydrophobia is? Rabies, in other words. These bacteria which cause this diseases. He studied them in great detail and identified all these bacteria, and he is for his great contribution to microbiology. He is called as the father of microbiology. So hats off to Louis Pasteur. Here comes the next great surgeon. He is a professor of surgery, Joseph Lister in Glasgow. So this great doctor, he applied Pasteur's work and he perfected them and introduced aseptic techniques in surgery. What are aseptic techniques? See, in the olden times, people, when they went to surgery, they were so enthusiastic, they would have just gone and operated on the patient without caring about or without having knowledge about bacteria and the need to wash hands or have asepsis. Asepsis means proper techniques by which one doesn't infect a patient during surgery. So people were unaware of the sterile techniques. This is where Lister came in and he identified carbolic acid as an aseptic agent. So using carbolic acid during any surgery with pus or anything, making it sterile, you're cleaning it up properly with carbolic acid, which disinfects and kills these bacteria. So what was dramatic was it led to less number of deaths. Mortality rate was reduced and the diseases, infectious diseases, post-operative infections came down. And that was Lister's great contribution to surgery, and he saved many patients from post-operative sepsis. Next comes Robert Koch. If you look at him, he was a German doctor, a practitioner who lived during 1843 to 1910. He perfected bacteriological techniques. What do you mean by that? For example, his assistant's wife, her name was Hess. She was, helping her husband, who was working in Robert Koch's lab, with an idea that they were having a tough time to solidify media, liquid media. How will you harden liquid media and make them solid? Like, for example, your jelly that you use, as for in shaving creams, how do you change the consistency, or as in paste, or anything which is between solids and liquid? She gave an idea about adding agar to the liquid broth, so it would set into a semi-solid medium, and then it becomes easier. So that idea, Robert Koch utilized it, and he perfected the technique to make semi-solid agars on which they can grow bacteria and study clearly the morphology of the colonies. The bacteria grows and forms colonies. They can study it and find out what was the causative nature of the disease-causing bacteria. And many diseases have been identified by this way. And he also studied the Bacillus anthracis, the anthrax-causing bacillus, and then he perfected the staining techniques. You see, bacteria per se do not have any color stains; they are colorless. But when you need to see it under the microscope, it needs to be fixed and then colored. Anything, you give them blue color, violet color, many colors. But how do you color bacteria, which are tiny organisms, real microorganisms? So they used aniline dyes. He changed the nature of the dyes used, and he experimented with that, and then there were bacteria were stained better, and it was able to people were able to, scientists even behind him, they were able to interpret them and study them better. So he was helpful to grow bacteria. With all these techniques, it was possible to grow only that bacteria which was required. That is called as a pure culture of bacteria. And the greatest discovery of all times, even till date we have benefited from that, is he discovered Mycobacterium tuberculosis, the TB causing bacteria, and Vibrio cholerae, the cholera causing bacteria. Only when you find out the villain, the causative agent, then you can find the device, the methods to combat them in this warfare against bacteria. So he had a great work he did by identifying these bacteria, and his next greatest help or guidelines to the scientific community is he, he devised Koch's postulates. What are postulates? They're nothing but a set of rules. Now, where was it used? These rules are used to prove that this bacteria cause this particular diseases. Nowadays, you know that any textbook will give you the causative agent of this disease. But please go back to that period when people did not even know that microorganisms were causing the disease. And the scientists had the great burden of proving that this particular bacteria caused this disease. So to prove it beyond doubt. So Koch's postulates help them. Look at this picture. So there are 4 important Koch's postulates. The first one being, there were the rules. Please observe carefully. I'll display the rules later. I want you to understand the concept behind Koch's postulates. Number 1, where any disease, that organism which causes the disease should be associated with that particular animal always. So you can see in number 1, this experimental animal, laboratory animal, rat has been taken. So there is a lesion near the ear. From this lesion, if you take out the sample and stain it and see it under the microscope, this bacteria should be present. It is present, right? Then number 2, the second rule is that this bacteria, so first you have identified and you have observed, seen the bacteria under a microscope and proved that this is present, associated with this animal in this disease caused. The second point is this: if you have to grow these bacteria in culture. You can see that number 2 is the culture plate, that yellow color, and those are the colonies. These bacteria look different under the microscope, but when they grow on the plate, you can visualize them. You can see them as big colonies. Looking at the colonies, and you do a staining of the colonies, and you prove that this is the causative agent which is still associated. You have been able to isolate or grow it in culture. That is what we call. This is the culture media, so you're able to grow it in culture. Number 3 is, if you take these colonies of bacteria, purify them, which contains the bacteria or any microorganism for that matter, and you inject it again into the experimental animal, it should reproduce and cause the same disease as in one. The same disease should come to this animal as well. The same infection, the same lesion should be reproduced in this experimental as well, animal as well. And once you get that same lesion, from that, if you recover that material and again grow it on culture media, you're isolating the organism again. You should prove it that it still is present again with this animal which has developed the same lesion, and you grow it in culture or you see it under the microscope. So you have proved it, that it has come a full circle, So this is valid proof that this particular microorganism is the causative agent for this particular disease. So these are Koch's postulates. They were first indicated by Henle. And then Koch went on to implement it. So number 1 is Koch's postulate: bacterium should be constantly associated with the lesions of the disease. That is what you saw in picture number 1. That number 2, it should be possible to isolate the bacterium in pure culture from the lesion. So think back again to that picture. Number 3, inoculation of such pure culture into suitable laboratory animals should reproduce the lesions of the disease. Has it done? Yes, it did. And number 4, was it possible to re-isolate the bacterium in pure culture from the lesion produced in that experimental animal? Yes, it was done. So this is valid proof that this particular microorganism causes this disease. Now, there is a fifth additional criteria to cross-postulate, is that also they need to prove that specific antibodies to the bacterium or the microorganism should be demonstrable in the experimental animals. Now, antibodies are produced by the experimental animal or human host, whatever. It is in defense to the microorganism which has entered. So, if you prove that the specific antibody exists, that is additional proof, postulate number 5. So, with this, people were armed with this. They had a new set of rules by which they can prove that, they can claim that this was the causative agent of this particular disease. Using these only, all the diseases and their infectious agents that we know have been attributed to them have been identified. Now, Pasteur and Koch had a lot of followers who took in their footsteps, and they have been honored with, they have contributed to the microbiological science by discovering many bacteria, and so many bacteria have been named after the discoverers, like Hansen, for example. People who discovered bacteria like leprobacillus, it causes leprosy, and we don't call any leprosy patient as such. We call them as Hansen's disease because it is a disease with stigma in honor of Hansen. And then gonococcus, a bacteria which causes a sexually transmitted disease, was discovered by Neisser. And staphylococcus, a pus-forming organism, and many other diseases was discovered by Ogston. Loeffler identified the diphtheria bacillus. He just identified the bacillus, but the toxin, if you see, the bacillus produces a poisonous substance-like thing, toxin. That was identified as by Roux and Ertzen. And the disease Malta fever, otherwise called as brucellosis, was named after Bruce, who discovered it. This is just a few. If you go and look back, study the literature, you will know that many such bacteria, there are hundreds of them, which have been named after their discoverers. That is your homework. Go on and find out how many other people have been named after the bacteria they have discovered. Here comes world famous experiment of Fleming. Now, this is a lesson for you people as well, where you need to keep your eyes and ears open and observe keenly whatever you look at. There might be an answer to a problem the world faced. Fleming was a scientist. One weekend, he went home. He was working on culture media processing staphylococcal bacteria, and he left it on the table and he went home. 2 days later, after the weekend, probably he came on that day and to discard away with a plate. That means to throw off these plates. He was surprised to see that there was a fungus growing. You can see this plate. The tiny organisms are the bacteria. The green colored organism which grows is a fungus. Now, he was about to throw it away, but then what did he look like? He was just observing it and he noticed that one particular thing. Do you see that? Do you notice that? I'll give you a few seconds to observe it. Everybody sees that, but only certain people think of answers like Fleming. Did you notice that near the fungus, that green colored thing, there were no colonies of bacteria growing at all? Something was inhibiting the bacteria from growing. And this surprised Fleming, and he was thinking something is produced by this fungus which kills these bacteria and doesn't allow it to grow. So what will happen if I was able to get it in pure culture? I can find a substance which can kill bacteria. And that's how Alexander Fleming went down into history, because he applied his mind to it. He discovered penicillium, that antibiotic substance produced by the fungus penicillin, which destroys staphylococcus, that bacteria. So penicillin is the antibiotic substance derived from the fungus penicillium, right? So Florey and Chain, the people after Fleming, Alexander Fleming, they discovered the great potential of penicillin, the antibiotic, to kill the staphylococcus. So they went on to produce in mass production. So this was said during World War I. So a lot of people were getting killed and dying due to infections, staphylococcal infections. They were saved by this miracle drug, penicillin. Now, Paul Ehrlich. You look at him, he looks like a scientist, doesn't he? He's a German person, and his great work is on toxins and antitoxins. No, he found out about the interactions. Remember the toxin we were talking about earlier, which were produced by bacteria. They are poisonous substance. Our body needs antitoxins to kill these toxins and neutralize it. This work he was doing. Now, so he was also known as the father of chemotherapy because of salvarsan, an antibiotic substance which was used. So this functions like the magic bullet too. Okay, so salvarsan was discovered by Paul Ehrlich, and he also had a bacteria named after him because he discovered it, Ehrlichia. And he also found out about the acid-fast nature of tubercle bacilli. Please remember, when you are studying tubercle bacilli, come back to this session and look about the acid-fast nature. Now, he also won a Nobel Prize in Physiology, and he went on to found the Paul Ehrlich Institute in Germany. Now, Ignaz Semmelweis. This person's story needs to be told, because in spite of his greatest contribution to microbiology and science and humanity, he was also, spent his last years in a mental asylum. He was branded and left there. A very pathetic story, but these stories also need to be told. When you delve into history, there are stories like this. He did good to humanity, but what did he do? Please go find out how he was instrumental in bringing down puerperal sepsis, just by hand wash techniques. Now the hand wash techniques that we all follow, it all started way back. With such a simple technique, Ignaz Semmelweis was saving so many mothers who had delivered the children in their puerperum. They had been saved from death by a simple technique of asepsis, hand hygiene. But it's a long story. To make it short, he was finally, his own peers did not agree with his views, and he became frustrated, and that's how he ended up in the mental asylum. And apart from these, there are a few important contributions, many important in fact, from other scientists as well. You may not understand the significance. I just want you to be familiar with these names. So you can come back, as you study microbiology, you can come back and look up at these names. Twort and de Herelle, they found out about the lytic phenomenon in bacterial cultures. Edward Jenner, we all should be thankful to him for he coined the term vaccine, and he was instrumental in finding out about the smallpox vaccine, the Jenner's famous experiment. For want of time, I'm making you familiar. Please go read those stories. Von Behring and Kitasato discovered antibodies which protect us so much. Baudet, he discovered the concept of humoral immunity, again with antibodies. Mechnikov discovered phagocytosis. method of cell-eating bacteria, and Jern's natural selection theory, Burnett's clonal selection theory, all have mentioned. Apart from these, there are many other people also. So, these great scientists have been recognized. Many of them have got recognitions by means of various awards, and the Dad of all awards, Nobel Prize, it has been given to many scientists, like from 1900s onwards. Emil von Behring for discovering diphtheria antitoxin, to almost 2015, quite recently, for William Campbell, who discovered that ivermectin can be used for filariasis, and UU2, he discovered artemisinin use for malaria, and these and the like were contributing so much to microbiology. And so at the end of this session, if you gain knowledge, that's fine. That should be the goal. And also, you need to concentrate on what are the possible questions they can ask you. For example, there may be questions on any of these scientists or their postulates or their contributions, which you need to be familiar with also. Thank you. So today, as we come to the end of this session, these great pioneers in microbiology, the world famous scientists who have contributed so much to humanity, they deserve a standing ovation. So here today, I encourage you to dream big, and one day you can even achieve the greatest prizes, the dad of all prizes, the Nobel Prize even. Thank you.